Hello, everyone. Welcome to the Livestock Heat Stress webinar series hosted by the USDA Agricultural Research Service. Thank you all for joining us today. My name is Maribel Alonso. We have a great webinar for you. Just a few reminders before we begin. During the webinar, you will be muted. If you have any questions, please select the Q&A option on your screen, and the presenters will respond to all of the questions at the conclusion of both presentations. All sections of this webinar will be recorded and available on the webinar's webpage. Now, I would like to introduce you to Dr. Steve Moeller, National Program Leader of the Food Animal Production. Dr. Moeller will be introducing today's speakers. Take it away, Dr. Moeller.
Thank you, Maribel, and all your colleagues at the Office of Communications. We appreciate your assistance in pulling together our webinar today. Our first speaker today is our research animal scientist, Dr. Jay Johnson. He'll be presenting on improving swine heat stress resilience through management, nutrition, and genetics. A little about Jay. He's a research animal scientist within USDA-ARS in West Lafayette, Indiana. He specializes in stress and nutritional physiology in that role. He received his bachelor's and master's degrees from the University of Missouri and his PhD from Iowa State University, and completed his postdoctoral training at Purdue. The overall goal of Dr. Johnson's research program is to identify production-relevant stressors and evaluate their impacts on livestock health, productivity, and welfare. Dr. Johnson's research program uses an integrative physiology approach, encompassing aspects of stress physiology, nutritional physiology, and ethology to develop and to improve our livestock husbandry practices, which can enhance our animal health and welfare while also maintaining or increasing our economic return for the producers. Specific areas of research include improving heat stress resilience in swine to reduce the negative effects of pre and postnatal heat stress and mitigating the effects of early life stressors on gastrointestinal function in pigs and dairy calves to improve health, performance, and welfare metrics. Thank you, Jay, for being with us today and presenting some of the work you're doing here within USDA-ARS, and I'll turn it over to you.
Well, thank you for that introduction, Dr. Moeller, and thank you for the organizing staff for putting together this webinar today, as well as the attendees. And so today I'm going to be talking a little bit about some of the work that my group here at the Livestock Behavior Research Unit has been working on over the past few years in the attempt to improve swine heat stress resilience through management, nutrition, and genetics. So we know that mean global temperatures have risen in the past century, and this means that animals are increasingly being raised in regions where heat stress is prevalent. And so the question remains is, what is heat stress? So how do we actually define it when we're talking about heat stress in our livestock? And so when we think about heat stress, we think about the balance between heat gain and heat loss by that animal. When we're in balance with heat gain and heat loss, both by heat gain through body temperature, the environment, and metabolic processes, and heat loss to the environment, that animal would be considered in thermoneutral conditions and would have a normal body temperature. However, when we get an imbalance in that heat gain to loss ratio, this is when heat stress can occur. For instance, when environmental temperatures go above that animal's critical temperature point, they can reach a state of hyperthermia and heat stress can occur. And when we add on other issues like metabolic processes, such as lactation or gestation in sows, the effects of that heat stress can be compounded. In an attempt to remain euthermic and maintain homeostasis, pigs will have a variety of different biological responses to that heat stress. And these are performed in an attempt for that animal to maintain survival, and it prioritizes survival over its productive state. Due to this, we see a variety of different production losses, things like reduced lean muscle growth and meat output per pig, reduced body condition, poor animal welfare outcomes, and in severe cases, mortality can occur. And due to this, we see a major economic impact of heat stress on U.S. swine production. Back in 2003, it was estimated that the swine industry would lose approximately $299 million per year. However, of course, this is quite old at this point, and considering the effects of both climate change as well as inflation, when estimating in today's dollars, this could be upwards of $481 million in losses today. In addition, another estimate said that there were approximately $55 in losses per sow per year due to reproductive issues related to heat stress. Again, this was based on 2010 estimates. And so when adjusting for that inflation factor, we see this number increase to around $88.55 per sow today. However, it's important to note that these values do not account for the postnatal effects of in utero heat stress on swine production. And so in the past decade, my group and others have been describing the negative effects of in utero heat stress on swine during postnatal life. We know that in utero heat stress, or IUHS, is associated with negative economically relevant postnatal phenotypes in pigs. The effects of in utero heat stress can be observed throughout a pig's lifetime, and we've seen that the first half of gestation generally elicits the most negative responses. A primary risk factor for in utero heat stress is elevated maternal body temperature, so either a chronic and low increase or an acute and high increase in body temperature. We see a direct piglet impact of in utero heat stress, things like reduced birth weights, greater pre-farrowing embryonic or fetal losses, reduced weaning weight, and lower survival rate of these piglets. However, we are interested in what is the future impact on offspring after they're born. And what we found is that in utero heat stress has a wide variety of negative postnatal impacts in pigs that occur during all phases of the swine life cycle and all phases of production. These can elicit negative responses such as a greater stress response in our animals, reduced growth rate, decreased feed efficiency, increased heat stress sensitivity, impaired reproductive performance, altered immune function, and decreased meat quality and quantity. And these all occur independent of the postnatal environment that these pigs are raised under. And so the question remains, how do we fix this? So what strategies can we take to mitigate both the negative effects of postnatal and prenatal heat stress in our pigs? And so our group has been focused on improving swine heat stress resilience through a variety of different ways. And so we define heat stress resilience as the ability to cope with and recover from a heat stress challenge. We take a three-pronged approach in looking at heat stress resilience. First, management, so husbandry practices and technologies to improve heat stress resilience. Nutrition, optimizing feeding practices. And genetics, or the development of thermotolerance breeding values. So first, touching on some of our work in animal management through precise environmental control. This is some research that had been funded by USDA NIFA, and we've conducted this research at all phases of production in non-pregnant and gestating sows, pre-nursery pigs, growth finished pigs, and boars. But today I'm really just going to focus on those non-pregnant and gestating sows. And so in this research, we're both looking at an animal's behavioral thermal preference as well as its physiological thermal stress response. And to do this, we've developed what we call thermoclines. And so they are two chambers that have a gradually increasing thermal gradient, and it allows the animal to actually choose what temperatures they prefer to be in and which temperatures they prefer not to be in. Taking this data, we can model it and we can determine what is the peak temperature preference of our sows. So in this case, we were looking again at non-pregnant mid-gestation and late gestation sows. And what we found is that our late gestation sows prefer to be at temperatures at approximately 14 degrees Celsius or 57 degrees Fahrenheit, whereas our mid-gestation and non-pregnant animals prefer to be at about 14.8 degrees Celsius or around 59 degrees Fahrenheit, which, if you look at the current Ag Guide standards, are at the lower end. In addition, we performed some physiological heat stress testing, looking at the body temperature and physiological responses of these sows at 3 different gestation stages to increasing temperatures. And with this information, we've been able to develop some heat stress thresholds. So defined as either mild heat stress, the point at which active heat loss attempts by that animal start, moderate heat stress, the point at which our active heat loss attempts fail and body temperature starts to rise, and severe heat stress, which is the point at which our body temperature can rise uncontrollably. And so what is the application of this data? So with this information, we've developed heat stress threshold equations, And so these are physiologically based heat stress thresholds that can predict when both respiration rate and body temperatures will go above thresholds based upon both ambient temperature and dew point. And these are differentiated by gestation stage to reflect the physiological changes that are occurring as gestation advances. We've integrated both our behavioral thermal preference and our physiological stress response into a decision support tool in collaboration with both Purdue University and the University of Illinois. With this tool, producers can input three different inputs into the thermal index: the reproductive stage of their animal, the ambient temperature, and the dew point. And they can get 6 different categories. That animal is either cool, comfortable, warm, mild, or moderate or severe thermal stress. Now, of course, we don't expect that producers will go through this each time they're trying to decide whether or not their animals are in a comfortable state. And so we have actually integrated this decision support tool into a smartphone app, which we call HotHog. Now, beta testing is ongoing, although if you are out there and you're interested in participating in some of the beta testing, you can reach me at j.johnson2@usda.gov. And this smartphone app is going to be targeted towards swine producers, but researchers performing heat stress studies may also find it useful. It can provide real-time alerts of heat stress risk levels, as well as management recommendations for producers. When you take a look at the app, we have both our login screen and our home screen. And looking specifically at the home screen, it'll give you your thermal index category, reproductive stage selection, so either open or early gestation, mid-gestation, or late gestation, thermal index hourly predictions, and a thermal index weekly prediction that includes both your index predictions as well as your daily temperature ranges, so you don't have to scroll back and forth to your weather application or this application to see what the temperature range will be that day. You can set your specific location, and at the bottom you can look at physiological outcome scenarios, barn management recommendations. You can build your own index, and you have your profile settings. In addition to management practices, we've also been doing some work looking at nutrition, specifically lactating sows and heat stress. We know that reduced feed intake is often cited as a primary reason for decreased sow milk production and litter growth performance during heat stress. And this is generally attributed to decreased energy available for lactogenesis or milk production. Because of this, there's been several nutrition and management-based mitigation strategies proposed, things like changes in feeding patterns, dietary supplements, or diet formulations. However, we are curious as to whether or not feed intake alone really explain that reduced milk production and litter growth. So in collaboration with Drs. Alan Schinkel and Bob Stwally, with support from Port Chekhov, we did some work with electronically controlled cooling pads developed at Purdue University. We know that they're effective in allowing sows to maintain normal body temperatures under heat stress conditions. They can improve heat conduction from the sow into the pad, and they're energy efficient. And so we combine these pads with our individual sow indirect colorimeters that we developed here at the Livestock Behavior Research Unit in order to look at individual sow metabolic heat production. And we use this as an indirect indicator of lactation output or milk production by these sows. What we find is that our animals that are cooled under heat stress conditions do have a greater metabolic heat production. And as we've seen in previous literature, heat production is closely associated with milk production. In addition, we observed improvements in piglet growth rate as well as weaning weights for our cooled animals. But despite the fact that we see these improvements in both milk output and litter growth performance, we did not really see any differences in feed intake. And so what does this mean? Essentially, we believe this means that increasing feed intake during heat stress may not completely rescue lost sow lactation performance, and that there's likely a direct biological effect of heat stress occurring on lactating sow performance, which is similar to work in dairy cattle that's previously been shown. And so feeding management should be coupled with effective barn cooling strategies to get the most benefit. Finally, the last area of work we've been working on is genetic selection for heat stress resilience. And this has been in collaboration with Drs. Luis Brito, Francesco Chiesi, and Christian Maltucca, with support from both Smithfield and USDA-NIFA. And so we know that heat tolerance is heritable, and selection for heat tolerance must consider both production and welfare relevant traits. We aim to develop thermal tolerance breeding value estimates and integrate these within a selection index for sows. In order to do this, we've been taking direct measures of swine heat stress under commercially relevant conditions during the summer months. We've recorded things like skin temperatures, body temperatures, respiration rate, body condition score, panning scores, cortisol, and in total, we currently have 1,645 animals with phenotypes and a little over 1,600 animals with genotypes. With this information, we've been able to develop heritability estimates. And what we find is that automatically recorded vaginal temperature is moderately to highly heritable across sows. However, the question remains is this a feasible measure for industry? Can we expect geneticists to go out and really measure, 1,600, 2,000, 3,000 sows at a time? Probably not. And so looking at this a bit closer, we've looked at time-specific vaginal temperature predictions. And so when we compare individual hourly measures of these sows against our 24-hour automatically collected data, we do find that our vaginal temperature heritability is relatively similar, and that the genetic correlations between individual hourly measurements are actually very highly correlated with our 24-hour data, indicating that you may be able to go out and simply take one measure at a certain time of day and get a similar heritability estimate. In addition, we were interested in whether or not some anatomical characteristics would be correlated with this vaginal temperature response. What we found was that both ear area and ear length were significantly genetically correlated with this body temperature response that we observed, as well as hair density. With this information, we've been able to develop genomic breeding values for heat tolerance, differentiating between both heat-sensitive and heat-tolerant animals. And we do find that the genetic merit of heat tolerance and sensitivity do differ. And so this means that selection is possible. And so some ongoing research from this is we've been doing some biological validation here at the Livestock Behavior Research Unit in West Lafayette, Indiana. We've selected 20 of the most tolerant and 20 of the most sensitive sows. This is based upon their genomic breeding values for heat stress tolerance and sensitivity. We're currently doing some intensive testing and phenotyping under these controlled heat stress conditions with the goal of identifying genes associated with both these positive and negative traits of our sensitive and tolerant sows. Again, to develop these thermal tolerance breeding value estimates for integration into a larger genetic selection index. Some follow-up work on this is we're looking at mitigating the effects of in utero heat stress through the use of genetic selection. And again, this is some work supported by USDA-NIFA. We are currently in year 2 of this project with some of the live animal testing upcoming. And so some take-home messages from today. Again, we know that heat stress has several negative effects on swine health, performance, and welfare. And this is because our pigs are prioritizing survival over productivity. We know that the economic costs of in utero heat stress may actually rival those of postnatal heat stress. They haven't yet been determined, but we know that the negative production and health effects of in utero heat stress occur throughout a pig's lifetime, regardless of season, and are not simply dependent on environmental conditions. And so improving heat stress resilience through management, nutrition, and genetic selection should be a priority for the swine industry. And as I've mentioned here, there are ongoing collaborative and multidisciplinary research efforts taken by the USDA. And with this, I'd just like to acknowledge all of our collaborators in this research, as well as Fast Genetics, Smithfield, and Mashoffs, and including our funding sources from the Pork Checkoff and USDA-NIFA. And with that, I can entertain any questions either now or after everyone has given the presentations.
Thank you, Dr. Johnson. And I think we will wait for questions. If you do have questions, we'll put those into Q&A and we'll follow up with those after our second speaker. But thank you again for a nice presentation on the work that you're doing there at the Livestock Behavior Research Unit. Our next speaker is Dr. Heng-Wei Cheng, and he'll present today on thermal perches as cooling devices for reducing heat stress in caged laying hens. Dr. Cheng received his MD from Medical School of Dongham University in China and a PhD from Wayne State University in Michigan. He joined the faculty at the Harbin Medical University in 1979 and the faculty at the University of Southern California in 1989, then relocated his program to USDA-ARS in Purdue and began studying mechanisms controlling stress responses in poultry. He received PSA's Poultry Welfare Research Award in recognition of his excellence and achievements in the research of poultry welfare in 2014. Dr. Cheng has educated over 40 individuals who hold positions at several universities and governments and privately funded research environments. He has been PI or co-PI on some 13 federal grants worth over $5 million and has published over 130 refereed publications in a variety of journals, including aggressive behavioral brain research, cells, Frontiers in Genetics, Journal of Complementary Neurology, Journal of Animal Science, Neuroscience, Nutrition, Psychoendocrinology, and Poultry Science. With that, I'm going to turn it over to Dr. Cheng to present again on thermal perches as cooling devices for reducing heat stress in cage laying hens. Off to you.
Thanks, Dr. Moeller and Maribel to put this together. I think that this is perfect timing. Right now, we all feel hot, so if we feel hot, the animal feel hot too. So also they gave a wonderful introduction about the heat stress and the heat stress related to damage. So because the principle used by swine and the poultry that's similar, so I just moved to stress in poultry, just avoid, to waste time. So I put some slides about it. So, but, maybe we start from here. Why heat stress is critical in poultry? So because chicken do not have sweat glands. Heating is a major measure for chicken to reduce body heat. Also, almost the entire chicken body is covered by feather. So this limited the capability for them to reduce body heat. Also said, like for higher production, so that means use more energy, so produce more body heat. Also, chicken has higher body core temperature with a narrow range, so that means chicken easily to be heat stressed, so also like in swine, so the heat stress chicken that's reduce feed intake or reduce energy used for production and so increase mortality. So this is current cooling measures quite popular used in poultry barns. One like similar to a sprinkler system, just spread water on the chicken and also reduce to the air, reduce the room temperature. Another is cooling pads. Warm air goes through the weighted pads, then become cold, and then get inside the room, so reduce room temperature. Another one is turn the ventilation system, use the fans through the air inside and then blow, through the room to reduce heat. So we just think about, is there another way to reduce, heat, from chickens? So these factors related to the study design. So one is chicken leg anatomy, so because chicken leg, so do not have muscle and do not have fat tissue, that's easier for chicken to release body weight. Also, there is a special circulation system in chicken leg. So the blood flow can be regulated, based on the temperature, so if the hot, more blood, through the leg, if cold, less blood. So 25% chicken body weight can be released through chicken's feet. Another the factor is roosting behavior, so because the commercial chickens, in this study, the hens still keeps rooting behavior, like the wild chicken, so, and also based on this behavior, so the Perch has been requested to for the arrangement case and the aviary system. Another factor, so is in the US, egg production facilities is changed from the conventional cage to cage-free based on more than 200 companies' demand, so because they announced only buy eggs from cage-free hens. So these are questions we ask is, so if the Perch inside the arrangement case or the aviary system can be modified to be cold Perch, and so to reduce heat stress in the hens. This is a team study, so, and supported by NIFA, AFRI, and my former PhD students, Dr. Hu, is a major person to conduct this study. This is a design of the cold Perch. So this we use a water chiller set up the 50 Fahrenheit and pump the water to water storage tank. Then use a pump, pump the water through the Perch, so it's level Perch. Also, we set up temperature sensor at a different level, cage level, chicken level, also input, output of the Perch, the line, so also we use one controller to modify the temperature change daily, so. This is one example how this system works, so around the 25 degree of the room temperature, and then we open the heater, so then the room temperature go up. At the same time, the Perch temperature go up. But if you look the supply line, the water temperature much lower than return temperature. So this means the chicken release body heat into the cold water, so then bring back to the water tank. So this means the co-perch the system can reduce the chicken body temperature through the chicken feed when they are roosting on them. So this is a study design. We include the entire life of the hens from day one, then until the molting, until 80 weeks, then goes through molting. So during this period of time, through 2 summers, so the heat stress 1 and the heat stress 2. One is at young age, one is at old age. Also the molting, so during the molting period, we also apply the heat stress. This treatment, we have the 3 treatments. So one is a cold perch, one is air perch. Air perch means all the same, the perch materials and the perch just without cold water circulate inside the perch, and the control just without perch. So, this case design, so that's we use one similar to the one used in the Dr. Hester study. So in her study, she report hens heavily used perch, and the perch did not affect egg production. Also, the perch can improve skeletal health of the chicken, So this is the floor space, perch space, and the feeder space used by per chicken. This based on the guidelines set up by Highlands. So this is the results. From the result, we can see cold perch hens had a lower core body temperature compared with another 2 treatments. Also, cold perch hens saw panting much late and at the low level, compared with another 2 treatment hens. So this means a cold perch can reduce, the heat stress effect on body temperature, and also effect on the heat stress behavior, panting in laying hens. Continue the results. So this saw the mortality. Cold perch hens had lower mortality compared to control hens, and feed intake. So the cold perch had higher feed intake during the stress, because one heat stress negative effect is reduced feed intake. But in this study, the cold perch hens maintain the feed intake, so they have the higher body weight compared with the hens from other 2 treatments. This is the egg production, so look the egg production, the hen's day egg production and the house production. So that's treatment, the significantly affect, production. So special, at old age, old hens, so cold perch hens had higher production than both, the air perch hens and the control hens. Also has heavier egg weight and the shell thickness. So this means the cold perch hens so eat more and absorb more calcium from diet. this saw some the data, of the hormones, so change. So this slide saw the cold Perch hens has higher, T3, the concentrations levels and but lower HSP7, so T3 is an active thyroid hormones involves in energy metabolism, so and this means cold Perch can reduce heat stress effect on hens metabolism. So his HSP70 that's is a kind of HISA protein. So the function of the HISA protein is protect cells from stress caused damage. So increase, the of the HISA protein 73. So that means more cell damage. So at here the lower, so his HSP70, that means cold Perch can reduce his heat stress caused, cell damage. So then after, receive this result, we saw how about induced molting? So because sometimes induced molting may conduct during summertime, that means combine the molting stress with heat stress. So this just saw the reason why need to do the molting, so because the benefits just through the molting cause regeneration and the regrowth of the reproduction system, then bring hens into a second laying circle, just by this extend their production life and improve egg production and egg quality, because after molting, hens lay larger eggs with high egg quality, and also reduce the number of the male chicks, to be killed, and the lower production cost. So in the molting, induced molting study, we found the cold Perch did not affect the pattern of induced molting, all the egg, so stop the eggs. And but during the molting, cold Perch hens, eat more feed. So there's a similar results, so we report during the heat stress. So, and also post-molting egg production much higher in cold Perch hens. So this means the cold Perch can reduce heat stress effect, on egg production, so. These outcomes, cold Perch can increase heat transfer from the chicken leg, to the cold Perch and reduce the core body temperature and increase or maintain metabolism. So through the feed intake and the thyroid function, hormone function, also reduce stress reaction. So through the hetero lymphocyte ratio down, and also reduce has lower HSP70 concentrations. So hetero lymphocyte ratio is a stress indicator used in the chickens. So also the increase increase egg production, performance, egg production, and egg quality, reduce mortality. Implication, just a co-perch can be used as an alternative for reduced heat stress in the hens. So, especially in the tropic and subtropic regions, but right now look like all the US, they're quite hard, So. In the future study, we want to see if the warm perch can reduce cold stress during the winter time. So the background is numerous studies have been conducted, to investigate effect of cold stress on the hens' health and the production during the winter time. So the question is if the warm perch can reduce cold stress in the hens. Implication, if this works, then the developed thermal perch system can be used for entire production, the period, so time, so that means not only used during the summer, also can be used during the winter time. So study design is similar to one reported in co-perch study, but at here we use the water heater set up at 86 Fahrenheit, then the through pump we make the circulation within the perch. And we also have the three treatment, the warm perch, air perch, and the cold perch. So these results may report, so later, so if there is a chance. So thanks. The questions. So.
Thank you, Dr. Cheng, for a very nice presentation on our poultry and our laying hens. Again, it is, as you indicated, time of year when we're facing heat stress across the country. So hopefully we can make some changes in our production systems to improve our productivity, both for our birds and for our pigs. Now it's time, I'll turn it back to Maribel and see if we have any questions from the audience.
Thank you, Dr. Moeller, and thank you, Dr. Johnson and Dr. Cheng for very informative presentations. As a reminder, please type your question in the Q&A box at the bottom of the page, and we will try to cover all your questions now. So I do have a question for Dr. Johnson that we got. Is it feasible to keep soil at their peak comfortable temperature range of 58 to 60 degrees Fahrenheit?
That's a great question. And so I would probably say not in the summer months, just given the sorts of, ventilation systems we have in swine facilities at this point in time. But unlike other thermal index systems, we wanted to make sure that we were also incorporating the comfortable range for what we see based on behavior. And so we think that this could be valuable, especially during the winter when we're putting a lot of energy into our facilities for heating. Maybe we don't need to heat them as much as we currently are, especially with our gestating or non-pregnant sows. And if we could drop our temperatures down a bit, into that 58 to 60 degrees Fahrenheit range, it could represent some economic savings for producers.
Thank you so much, Dr. Johnson. Great answer. We just got another question. Why is the mortality was higher in cooler perches group hens? I guess that's for Dr. Cheng.
In my study, that's in the case cold hens, that's lower, not higher. So yeah, because the cold perch can reduce heat stress, but another two groups, special control hens, so they do not have any facilities to use. And if the room, because in our study room set up 32 to 35 degree that's really quite stressed for the hens. So yeah, that's a cold perch has had lower mortality than control hens. But the air perch hens, the mortality close to cold perch hens, because they still have perch, then they can move around, So also when they stay on the perch, help them to release body heat, so because that has more body surface, to release heat, so but control hens do not have any chance. They only on the floor, but that's the top floor hens and the heads, the middle and the lower hens below them. So that's also body heat just come to them. So yeah, but in my studies, cold hens had a lower mortality than the control hens. Thank you.
Thank you, Dr. Cheng. I do have another question for you. What is the economic expectation based on the inputs and outputs of your research?
Oh yeah, that's really good questions. So because we just did a small study, so based on the data we collected, so that's from the Dr. Wetmer group, paper published, so that's based on the input and the output, so that's because reduce mortality and the increase egg production, that really can reduce the input to set up the perch. So this kind of based on data collected from the mass study. But the second one, so because we know the poultry production facility is changing from conventional cage to cage-free. So the benefits may be increased, with the changing, because when modify the product, set up new facilities, at that time can put this system in, so that's really reduce input, labor and the materials. So yeah, bottom line, based on my study, current study, that's really can reduce the perch, the input, the cost, but with the future maybe reduce more, so with the changing, Thank you.
Thank you. We hope for that. I don't see any more questions. If you do have any questions or comments for Dr. Johnson or Dr. Cheng, please feel free to submit your question through the Q&A option at the bottom of your screen. Don't see any more questions. So for now, if you do have any questions after this webinar, feel free to send them directly to Dr. Cheng or Dr. Johnson. Their email address are on our main website for this webinar. I'm going to add the link on our chat option here on the webinar. Feel free to send it to them, or you can send it to me, maribel.alonso@usda.gov, and we'll be happy to answer any questions that you may have after this webinar. Thank you for joining us today. Please remember that our next section is on August 3rd from 2 p.m. to 3 p.m. Eastern Standard Time. And our link will be available on our main website. I'm going to go ahead and join the link with you. And also these webinars will be recorded and also available for viewing later on demand at our YouTube channel or on the main website for this webinar. Thank you so much, everyone.
Thank you.